So we examine now the muscular system module from a microcosm standpoint. So now we're not looking at these very big muscles, the deltoid muscle and the biceps brachii and the soleus muscle. Now we have to look at the small muscle fibers and how does everything work. So muscles, when we look at a muscle cell under the microscope, what do we see? Well, we agreed in the lab that there are three different types of muscles. There's a cardiac muscle, and there is skeletal muscle, and there's also smooth muscle. Well, those three muscles, they are muscles in the end of the day, but what they have in common is that they are excitable. So all of them are muscles, meaning all of them have to have something in common. And they have to have different things. That's why they are different. So what is in common between all muscle cells? The fact and the property and the characteristic that they are excitable. You're not a muscle cell unless you are excitable. And what is the difference between the cardiac muscle and the skeletal muscle and the smooth muscle? And that would be that some of them are under voluntary control, like the skeletal muscle, your biceps, your triceps. You are able to move your bones because when these skeletal muscles get contracted, they are able to move the bone. But cardiac muscle is not under voluntary control. It's under involuntary control. You move your arm, but do you move your heart? Do you move your heartbeat? Do you control your heartbeat? So the heart muscle is under involuntary control. And the smooth muscle for vasoconstriction and vasodilation and decreasing the radius and increasing the radius, modulating the blood flow rate, all of that is important and it is under involuntary control. But nonetheless, whether the three types they are different, the three types of the muscle tissue, they are different in that some of them are smooth muscle, some of them are skeletal, others are cardiac. In the end of the day, when we look in the microscope, we have to see things in common. So you see these big proteins and you see these holes. Please, you see where my mouse is. We're going to learn today what is the value of these holes, these cavities. You see, this cavity is the site where you see this golf head, you know, the golf head. So this golf head is going to enter into this cavity and it's going to cause the contraction. So we know that our muscle cell is when it gets excited, it will contract. The muscle cell, after getting excited, it will get contracted. So the contraction happened with this cavity. We're going to learn more about it. So in order to be a muscle cell, you have to have five characteristics. Number one, contractility. So the muscle cell has to be able to contract upon excitation. Excitability is the property that enables the muscle cell to propagate and to extend an action potential. And then there is conductivity. Conductivity is very important because it gives the skeletal muscle cells the property of being a syncytium. A syncytium means that the action potential is going to flow through the gap. Remember, we talked about gap junctions, these holes through which the solutes are going to flow from one cell to the next cell, and the electrolytes are going to flow from one cell to the other cell, and also the action potential. So you see in your heart, your atria contracts at the top, and then they relax, and then your ventricles contract, followed by their relaxation. And the elasticity, that is a structural component. A structural component of your muscle tissue is that it is elastic, it is malleable, it is not rigid like the bone. No muscle cell is going to be rigid because it doesn't have what makes the bone rigid and strong. It is the cartilage, the cartilage. Epithelial tissue can sometimes be strong due to the keratin, the presence of keratin. So let's begin with contractility. Contractility is the ability of the muscle proteins to come together, to shorten. When the sarcomere shortens, what does contractility actually mean? They're running around. When they get older and take A and P, they're not gonna run around. Okay, so what do we mean when we say shorten? So the muscle cell will shorten. I have a dream one day they will have nothing like this. Okay. The blue one. Hmm? The blue one. Okay. It's not gonna work too. The green. No, I'm tired of this game. Okay, sarcomere. Okay, this one is working. Sarcomere. So the sarcomere extends from the Z line to the Z line. If your attitude, if you just want to go and look at the back of the question in the open stacks, you're gonna see that now from now on. You're gonna have to pay attention to class, to what the questions that I'm teaching and the things that I'm doing in class. So pay attention because I'm gonna ask about those too. So from the Z line to the Z line, that is the sarcomere. The sarcomere, upon contraction, what's going to happen? Are we going to have wider Z line to Z line, or are we going to have shorter Z line? So the distance between the two Z lines, are they going to, when we're contracting, are they going to shorten up or are they going to lengthen? They're gonna shorten. Excellent. So the ability to contract is the ability to make the sarcomere smaller. And it means the shortening, but more accurately, increase in tension. And a muscle does not necessarily shorten, but it is the sarcomere that comes closer. So you're not gonna, when your skeletal muscle contracts, you're not gonna get smaller. You're not gonna shrink. But it is the sarcomere that's gonna have a lot of tension. And then excitability. The excitability is the ability to respond to stimuli. So the stimuli may be a chemical stimuli, maybe a mechanical stimuli, or a electrical stimuli. Remember, our muscles, they contract. Sometimes if you touch the hot stove or they put ice on your back, you contract. So that's an example of a stimuli. That hyperthermia, that's hypothermia, that's cold weather, that's really hot weather. And then you have electrical signals. These electrical signals mediates. These electrical signals are passing. These electrical signals, they are mediated and they, for example, like an electric voltage. If you get hit by electric voltage, your muscles are going to contract. You know, they used to have the electric chair for execution. So that's what happens. They supply so much electricity, hyper voltage, that's going to cause severe contraction and terminal, irreversible, stressful damage to the sarcomere. Yes. So is that why, like, when your body gets sick, when you're electrocuted, and it's like, yeah, when you're cold, when you're hot, all of that. And also chemical. So your muscle tissue is gonna be affected by chemical stimuli. How is that? Anesthesia. So the anesthesia is going to cause the contraction and the South Indian native tribe, they throw the blow dart, and the blow dart has a chemical curare. Curare. This curare, this curare is going to hit your foot, and you can't walk. And then they're gonna take you and eat you. Oh no. Oh yeah. It's called cannibalism. Yeah. They still do it up to today in these Amazon jungles, and many tourists go there for sightseeing. They end up being eaten alive. Okay, the conductivity is the ability of the electrical changes to move or propagate along the plasma membrane, and we talked about that. we talked about how the influx of sodium through the channels. You see, these are the channels, and this is your plasma membrane, and these are your phospholipid heads, and these are your tails. Okay, so this is the middle butter. This is the middle non-polar segment. That's the head. So in order for depolarization to happen, sodium has to flow inside. You must have an influx of sodium from an area of a really high concentration of sodium to an area of a really low concentration of sodium, and that is diffusion. That is the movement of diffusion. So the influx of sodium is going to cause, if we look at this very important figure here, we will see that on the y-axis, we have millivolt. That is the electricity. And on the x-axis, what do we have? We have the time. So as the time moves on in milliseconds, the voltage increases. Okay, so when sodium influx, the body is in a resting state known as negative resting membrane potential, and it's at negative 65 millivolt. When sodium enters, that is known as depolarization. When sodium enters from outside to the inside, that is known as depolarization. And depolarization causes an increase in membrane voltage until we hit a peak. So this is called depolarization. And when we hit the peak, another type of channels are going to open, and these are the potassium channels, allowing now the efflux of potassium from inside, because potassium is a lot inside, unlike sodium, who is a lot on the outside, a little bit on the inside. Potassium is a lot on the inside and a little bit on the outside, and that is going to cause the repolarization or the hyperpolarization, and so on and so forth. We're going to oscillate, go up and go down. When we go up, sodium influx, and when we go down, deflux, efflux of potassium. Excellent. So when we go up, there is an influx of sodium. When we drop down, there is a efflux of potassium. So that's the conductivity. Extensibility is the ability of the muscle cell to stretch up to 3 times its resting length without being damaged. So that's the extensibility. Elasticity is the ability of, when you stretch, you get longer and your tissue doesn't rip apart. If you try to extend the belt so long, the tension is going to be so it's going to rip in the middle. But your muscles have the ability to extend for really long periods, as much as 3 times as long as its normal size. So the elasticity is the ability of the muscle cells to return to the original shape after being stretched. So after we stretch, we recoil. So elasticity is the ability to recoil and return back to the normal length. So the structure of the muscle cells. So most of the muscle cells are with the same organelles as any cell, with some differences. So obviously we said that every tissue was comprised of different types of cells. Every tissue is comprised of different types of cells. Why? Because the cells have different functions, they have different physiology, therefore they will have different anatomy, they will have different structure. So the muscle cell, also known as the myocyte. So let's look at now at the myocyte. The myocyte is comprised of cells Okay. So what's gonna happen is you have the muscle fiber F, 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 muscle fiber F. Each muscle fiber is wrapped by a fibrous capsule known as the endomysium. That's the endomysium, blue endomysium. So the endomysium is the raincoat that the muscle fiber has. Then multiple muscle fibers are wrapped around by the perimysium. So the perimysium wraps them around. Then multiple perimysiums that have muscle fibers. Okay, then all of them are wrapped by a third layer that surrounds all of them, and that is known as the perimysium. So there are fibrous capsules that wrap around our muscle fibers. There is one fiber that encapsulates one muscle fiber, then multiple muscle fibers are encapsulated by a one layer, then the entire myocyte is wrapped. You see, this is the outside tissue. So why do we have a lot of nucleus? Why do we have a lot of nucleus in our skeletal muscle tissue? Because the nucleus is where we have the DNA. And when we have the DNA, we need the cell division cycle. Remember, the cell cycle has two phases. What are they? What are the two phases of the cell cycle? I'm sorry? There is interphase, and mitosis. So interphase is divided into three parts. Number one, you have G1 and the S and G2. While mitosis is going to have prophase, then metaphase, then you have prophase, prometaphase, then metaphase, then anaphase, then telophase, then cytokinesis. So the ability to spin the cell cycle is gonna burst and give us a lot of tissue, a lot of cells. So we use that for healing. So if we have tissue that has been damaged, we need to remove it and build up tissue in its place. We go spin the cell cycle. Or if we are growing up like a child in development, during development, so we develop and we spin the cell cycle in order to grow in the size. So the cytoplasm of a muscle cell is known as the sarcoplasm. So the cytoplasm is known as the sarcoplasm, and the plasma membrane of a muscle cell is known as the sarcolemma. So a muscle cell plasma membrane. So the plasma membrane of a muscle cell is known as sarcolemma. And in addition to that, your plasma membrane is the sarcolemma, and the cytoplasm is the sarcoplasm. So the sarcoplasm is the same thing as cytoplasm. Cytoplasm, sarcoplasm. Yeah, yeah. The smooth endoplasmic reticulum is modified into a sarcoplasmic reticulum that stores calcium. So we don't have an endoplasmic reticulum in the muscle cell. We have a sarcoplasmic reticulum that is made up of cisterns. Cisterns. That is made up of cisterns. These cisterns are full of calcium. So when the action potential gets to the muscle tissue, this cistern is going to open up and all the calcium is going to come out. It is the action potential that's going to make the cistern open up and all the calcium is going to come out. Where is the cistern? It's in the sarcoplasmic reticulum. The cistern is in the sarcoplasmic reticulum. And the sarcoplasmic reticulum is going to allow all the calcium to efflux. So the smooth endoplasmic reticulum is modified into sarcoplasmic reticulum that stores the calcium. So there we go. That is the sarcoplasmic reticulum. That is the sarcolemma. And that is the sarcoplasm. That is the sarcoplasm. The myofibrils make up about 50 to 80% of the cell volume. And these are bundles of specialized proteins. And the organelles are found packaged between the myofibrils. So these are the myofibrils. So inside each myofibril, what do we have? We have the microfilament. So we have multiple microfilaments surrounded by all of them, they make up the microfibril. So that is the myofibril, and these are the microfilaments. So you see these tubes, if I pull one out like spaghetti, so it will come out really long tube, and this is your myofilament. What is the sarcoplasmic reticulum full of? I'm sorry? Yeah, it's full of the calcium cisterns. And when an action potential comes, what is that going to cause? Excellent. And the efflux of potassium we're going to see is going to cause the contraction. We're going to see in a little bit. So the structure and function of the skeletal muscle fiber. So the muscle cells differ from most cells. There we go. We've seen in the lab that this is a structure of the muscle fiber. It has a lot of nucleus. You see, it has a lot of nucleus. 1, 2, 3, 4, 5, 6. You see all these nucleus. Why do we need the nucleus?
Because of his DNA.
And why is the DNA important? For? Yeah, for the cell cycle. So we need to, when the skeletal muscle tissue is damaged, we need to make new skeletal muscle tissue. Okay. It could be up to 30 centimeters long, and it is striated, and it has multiple nuclei, multinucleated. Why is it striated? Because of these dense microtubules, these cytoskeletal proteins that look very dark under the microscope because they are important for. Remember, I showed you when we began the lecture the hole and that golf head? These are the striations. You're not going to see those a lot in a neuron because it doesn't have to contract. You're going to see it here because it has to contract. So, structure and function of the skeletal muscle fibers. They are composed of many cylindrical myofibrils surrounded by a sarcolemma. So, you see you have multiple myofibrils, and the myofibrils are surrounded by a sarcolemma. There we go. So, that shows us a muscle fiber. That is a muscle fiber. If you pull out, a muscle fiber is made up of multiple myofilaments. Remember, we talked about that. And these are the terminal cisterns of your sarcoplasmic reticulum. So this sarcoplasmic reticulum is going to throw out a lot of calcium, and the calcium is going to bind to the myofilaments. This entire unit is your muscle fiber. And you see the endomysium, that is endo from the name from inside, endo. So it is the layer. You see, it's like a capsule, like a coat. It's like a raincoat. It is covering. What is it covering? It is covering the muscle fiber. What is the muscle fiber made up of? Many myofilaments. So you have myofilaments, and the myofilaments are all covered by the sarcolemma, and on top of the sarcolemma, you have the endomysium. Okay, these are your cisterns from your sarcoplasmic reticulum, and they secrete. What do they secrete? They secrete calcium, and the calcium causes the contraction. We will see how. What are these transverse T tubules? What are these transverse T tubules? These T tubules are what the action potential is going to flow through. So when the action potential is flowing down the T tubules, when the action potential is flowing down the T tubules, that's going to cause the calcium to be secreted from these cisterns. And when the calcium binds this muscle tissue, that's going to cause contraction. So what's the function of the transverse tubules? Nope. They mediate the action potential through the tubule. So the action potential passes through these T tubules. They are conduits. They are passageways for the action potential. And then when they pass through the tubules, then they're going to cause the cisterns to release calcium. Okay. The sarcolemma. So what does the sarcolemma do? The sarcolemma forms transverse tubules that enter deep into the muscle. The transverse tubules are adjacent to sacs of the sarcoplasmic reticulum called the cisterns. You see, the terminal cisternae. Now it makes all sense. Why are these transverse tubules so close to the terminal cistern? Why are these tubules so close to the blue? Why are these transverse tubules so close to the blue terminal cistern? Yeah, because they mediate the calcium, the action potential. Then the action potential is going to flow from these T tubule into the cistern. And then out of the cisterns, the calcium is going to flow into the muscle. The transverse tubules are adjacent to sacs of sarcoplasmic reticulum called the terminal cisternae. Okay. These are openings of the T tubule. This is a nucleus. The nucleus, and that is your mitochondria. Why do we have to have a lot of mitochondria? Because the mitochondria provides ATP, energy. When you say energy, that's very vague because there is electrical energy, thermal energy, kinetic energy. It does not provide all of that. It only provides chemical energy in the form of ATP. So now we're going to see the involvement of ATP in the muscle contraction. In fact, ATP is important for muscle relaxation. That's why people who are dead, they have something known as rigor mortis. What is rigor mortis? Yes.
It's when the body goes stiff.
Yeah, it goes really stiff. This excessive stiffness of the body doesn't happen in a normal person. Why? Because we have ATP. So ATP is gonna relax your body from the stiffness. But the problem is, the dead people, can they make ATP? They cannot make ATP. Why can't dead people make ATP? Why can they make ATP? Yes, the cells are dead, but what are the dead cells no longer able to do? And that is,
It can't do it.
so do we need energy for mitosis? Mitosis is for dividing of the cells. So we are stiff. It doesn't mean that the cells die. We are stiff. It doesn't mean that the cells are dead. It means that the cells are in a terminal state of locking and contraction. We need to relax them, and they only relax with ATP. So the cells are dead. They cannot provide ATP, and that's why we cannot relax the muscle. But what are the things that must be done to get ATP that the cell is no longer able to do because they're dead. Metabolism. The cells that are dead cannot perform metabolism, namely catabolism. They cannot break down carbohydrates to get ATP. They cannot break down lipids to get ATP. They cannot break down nucleic acids to get ATP. They cannot break down carbohydrates or proteins to get ATP. Therefore, we cannot make ATP and then we cannot relax. But ATP is not important for contraction. That's why the dead bodies are contracted. The ATP is important for relaxation. And we will see that. But for now, it should be no surprise that our muscle tissue is full of our muscle tissue is full of ATP because when we are alive, we need a lot of ATP from the mitochondria that's going to go to the muscle cells and cause what? The relaxation of the muscle. Excellent. Okay. There's a set of transverse tubules and two terminal cisternae that forms a triad. Triads are important in muscle contraction. That is the triad. It is made up of transverse tubules and two terminal cisternae. You see the two terminal cisternae. Why do we have terminal cisternae? So the action potential can come passing by in the middle, causing the release of calcium. Structure of the myofibrils. The myofibrils are composed of many filaments. They are composed of many filaments. So the openings of the T tubules, these are the openings of the T tubules, and you have sarcoplasmic reticulum, and these are transverse tubules, and these are terminal cisternae. You have myofibrils, and the myofibrils are made up of, these myofibrils are made up of myofilaments. So the myofilaments comprise for you the myofibril, and the myofibril, multiple myofibrils are going to give you the muscle fiber. So you have this whole big cylinder is made up of tiny spaghettis. Those tiny little cylinders inside are your myofilament. The myofilament or the microfilaments make up for you a myofibril, and many myofibrils encapsulated with the sarcolemma and then encapsulated with their additional endomysium. That's going to be your muscle fiber. So each myofilament consists of different proteins. There are contractile proteins, proteins that are actually engaged in the process of contraction. These are the proteins that shorten the sarcomere. And there are other regulatory proteins. And these are the proteins that regulate the contraction, but themselves, they do not contract. And there are structural proteins that hold the myofilaments in place and they maintain the stability of the microfilament. So let's look at them. So you see, look at here. What do you see? Remember the golf head? That is your myosin. So the thick filaments are made up of myosin. Thick filaments are made up of myosin. What is myosin made up of? You see the two golf heads? That is myosin heads. And look at the paired tails, braids. These are the tails. So you have two golf heads with two tails that are wrapped around each other. And that is your thick filament. That is your thick filament. And then you have the thin filament. The thin filament is made up of actin. Actin is the thin filament. You see, that is your G actin, the purple. You see the hole? So it is thin. Excuse me? No, but you say you're so loud. Like you have to respect. This is unbelievable. This is even caught in the microphone. No, you did something. You went, oh, like that. Have respect, please.
But I'm not bothering you though.
Yeah, you did. It's so disrespectful. You yawn like that.
Alright.
Please have some mannerism and professionalism.
I have to do my thing.
What are our thick filaments made up of? Two golf heads and two braided, intertwined tails. So that is your thick filament. So on the exam, our question is going to be your thick filament is made up of actin or myosin. So look, these are the myosin heads. Do you see the myosin heads? So the myosin heads make up the thick filament. Myosin thick. And what is that? That is actin. So actin thin. Actin thin. Actin thin. Actin thin. Myosin thick. Myosin thick. Actin thin. And not just by the name, by the structure. Does actin have two golf heads? Does actin have two golf heads? Who has two golf heads? Who has the two braided intertwined tails? That's why it's thick. Now it makes sense because you have double of them and they are intertwined. These are thick. And then you have the thin filaments. These are made up of actin. And actin has the hole inside it. So you have to know that one. And these two, the red two golf heads, these are your thick filaments. And the purple actin, these are your thin filament. But look, that's not all we have. We have tropomyosin and troponin. You see tropomyosin, the long rope, tropomyosin, and you see the yellow troponin? These are not contractile proteins. These are regulatory. These are regulatory proteins. You see the regulatory proteins? That is myosin and troponin. You have to know that. You have to know that the red thick filament myosin, that's contractile. And the blue hold actin, that is contractile. But troponin and tropomyosin are not contractile. They are regulatory. So you got to know what are your two contractile proteins? Actin and myosin, regulatory. And what are the two? I'm sorry, these are contractile. These are the ones that contract. Remember, the golf head goes inside the hole. You see, the golf head goes inside the hole to make the contraction. But these guys, troponin and tropomyosin, they're not contractile. They are regulatory. Excellent. Okay, thick filaments. Now let's look at the thick filament. Thick filaments are composed of contractile protein myosin. And you have myosin that has two globular heads and two intertwining tails. You see, these are the two heads. You see the two golf heads and the two intertwined tails. The two heads and the two intertwined tails. These are myosin heads, and these are the myosin heads, and these are the intertwined tails. That is what is that, guys? What is that here? That is the thick filament because it has your myosin heads and the intertwined tails. Myosin head and intertwined tail. And this is the neck. This is the neck. So you have the two golf heads, the neck, and the two intertwined tails. And that is the thick or thin filament. That's the thick filament. Okay, now thin filament, it is composed of three things. It is composed of actin, tropomyosin, and troponin. Actin, tropomyosin, and troponin. You see, this is your golf heads, the neck, a hinge, and then the two tails. And then you have actin, tropomyosin, and troponin. So you see, that is actin, tropomyosin, and troponin. This is troponin, this is tropomyosin, and this is actin. Okay, it is a rope-like regulatory protein that spirals around two actin strands. You see, two actins, and that's the tropomyosin, and that is the troponin. Okay, please pay attention. This is really, really important. When the calcium, remember we talked about how the action potential, you have a lot of, membrane voltage change that causes the action potential to pass through what kind of tubules? What kind of tubules? Transverse tubules, excellent, or T tubules. Then the T tubules are going to carry the action potentials to the cistern, Then the cisterns are gonna secrete a lot of. That's where the calcium is gonna go. The calcium is gonna go to the troponin, drag troponin down. So you see the holes. The holes are not exposed, so your myosin can bind to them. Who's covering them? Who's covering the holes? You see the holes are covered. So myosin is what the golf head. The golf head cannot go in the hole. And then the golf head cannot go in the hole because it is covered by myosin. And then when calcium comes, it's gonna bind troponin. When calcium binds troponin, troponin is gonna drag and remove the tropomyosin away, and then the hole is gonna be open. When the hole is open, who's gonna bind to it? The head of the good, very good. The head of myosin. Myosin. That's why you gotta go home and review it. But the idea is there. So calcium is gonna bind troponin. Troponin is gonna drag the tropomyosin away, and then you're gonna have the active site, and then you're gonna have all of the cocking movement between the myosin golf head and the active site of the actin. That's why they call this one actin is a contractile because it contracts. Myosin is contractile because the golf head goes inside. But these two guys, troponin that cover the active site, these are your regulatory. So where does calcium bind to? Where does the calcium bind to? Troponin. And when calcium binds troponin, what happens? Excellent, excellent. It moves tropomyosin. You see, who's covering the active site, the hole? Who's covering it? Mitosis? Myosin. Tropomyosin. Myosin is going to bind to the active site. It is tropomyosin, the long belt, that is covering it. Okay, troponin. You see, that's troponin. Can you recognize troponin? Troponin is a second regulatory protein holding tropomyosin in place. Tropomyosin and troponin help switch on and off, contraction. So you see, tropomyosin and troponin help switch on and off the muscle contraction. That is very important. It's very important to know that calcium binds troponin, and then you drag that down. You're gonna expose the active site on which the myosin is going to bind. Okay, elastic filament. Elastic filament you would have is composed of large structural protein called titin. So this titin, this is very important. So I'm going to ask you on the exam, what causes the structural myofibril to be elastic? So this is no longer we have the protein elastin. like in our bone tissue that makes the bone a little bit malleable at the joint when you have the articulation. This is not the bone no more. This is the skeletal muscle cell. So it is composed of large structural protein called titin. So what is the job of titin? What is the job of titin? Titin is important for the elastic movement. Yes, it uncoils when stretched and recoils to its original shape. It uncoils when it's stretched and then recoils back like a spring. You know, the spring, you stretch it and then it recoils back. And it assists in the elasticity of the muscle. It runs through the core of the thick filament to stabilize the myofibril. It runs through the core of the thick filament to stabilize the mind. There we go. What is that? That is the titin. What is the job of titin? Its job is elasticity. Excellent. And recoil. Elasticity and recoil. Remember we said our muscle can stretch as long as how long its original resting length? 3 times. Very good. So these are all exam questions. So for example, multiple choice. One of the characteristics of the muscle cell is extensibility, meaning that the muscle fiber is able to extend as much as ... its original length. Once, twice, 3 times, or 4 times. 3 times. Now, if you were playing on the phone or daydreaming, that point skip you, and then you're gonna get that question wrong on the exam and on the quiz. So you're here, invest your time. Don't be you come in here, then you go home, you listen to the lecture again. So be here one time, focus, and you don't have to do the job twice. So you got to go home and focus on the main point, but you don't have to repeat the lecture again. So now let's look at that. We said, what is a tendon? A tendon always connects muscle to bone, and a ligament always connect bone to bone. So you have a muscle, that's a muscle, that is a muscle, and the muscle is made up of multiple fascicles. Fascicle, that's a 1 fascicle, 2 fascicle, 3 fascicles, 4 fascicle. The muscle, the big muscle, who's wrapping? What is the fibrous capsule? What is the bag? This plastic bag that surround. What is this plastic bag that surrounds the muscle? What is it called? The epimysium. That's the one all the way on the outside. Then when you move inwardly, you have multiple fascicles. Each fascicle has what surrounding it? The perimysium. The perimysium surround the fascicles. Then when you pull out each muscle fiber, you see that the muscle fiber is surrounded by the endomysium. So you have muscle fiber that is made up of myofibrils and so you have myofilaments that make myofibrils, and the myofibrils give you a muscle fiber. Around it is your endomysium. Then many muscle fibers give you a fascicle surrounded by the perimysium. And then all the fascicles give you a muscle surrounded by the epimysium. This is very important. So you have your very important microfilaments. You have very important. These are myofilaments. Multiple myofilaments will give you a myofibril. Okay? And you see that so you have myofilament, then you have myofibrils, and multiple myofibrils are surrounded by multiple myofibrils are surrounded by your endomysium. So you have multiple fibers. So the multiple fibers will give you a muscle fiber. And then the muscle fiber, this one, this muscle fiber, you have myofilaments. Many myofilaments giving you a myofibril. Multiple myofibrils will give you an endomysium. So that's the inner layer. Endomysium surrounding the muscle fiber that is already made up of multiple myofibrils, that is made up of multiple myofilaments. Then you have multiple muscle fibers to give you a fascicle. And what surrounds the fascicle? That is the perimysium. The multiple muscle fascicles surrounded by the perimysium are all encapsulated by one really big coat, one capsule from outside, known as the epimysium. Known as the epimysium. The sarcomere. The sarcomere, that is the contractile unit. So if I ask you on the exam, what is the contractile unit of the muscle cell? It is the sarcomere. It is a made up of thick and thin filaments that form alternating light and dark bands in the sarcomere. There we go. Do you see the Z disc that we began with? We said that Z disc, these Z disc, they contract, meaning that you see how the sarcomere, what is the length of a sarcomere? From one Z disc to the next Z disc. That's an expansion. So that's one Z disc. You guys see the Z disc? One Z disc to the next Z disc, and between them is the sarcomere. Now, do you see the blue? Who's my blue? The blue that has a hole in the middle, made up of balls. That's actin. Very good. And what is the red golf heads? Myosin. That's why the golf head go inside the hole of the actin. But it cannot do that now because who's covering it? Who's covering it? Tropomyosin and the troponin. But when calcium come in, it's gonna remove it, and then the golf head is gonna go perform the contraction. So now, when we look histologically in the microscope, we see that there are thin bands and there are thick bands. That is a thick A band, and these are thin I band. The I band contains only thin filaments. So the I band has thin filaments. The A band contains the entire thick filament, and there is a zone of overlap that contains both thick and thin filaments. There's an H zone. That's an H zone in the middle. That's the H zone in the middle. The M line is the dark line in the A bands. And the M line has proteins that hold thick filaments in place, and they anchor the elastic filaments. So now you see, who is my thick band? Who is my thick band? The A band is the thick filament. And who is my thin filament? That is the I band. And the H zone is a zone that contains only the thick filaments. Just remember that the sarcomere is how you have the shortening of the Z disc. You see the Z disc? That's a Z disc. That's a Z disc. When it gets shorter, what happens to the sarcomere? It gets shorter too. And this is the actin or myosin. That's myosin. Is this actin or myosin? Actin. Actin and myosin, are they regulatory or contractile proteins? Contractile. What about tropomyosin and troponin? If we begin with tropo, whether tropomyosin or troponin, in the end, it is not contractile protein. These are regulatory. Excellent, excellent. Okay, so what does the sarcomere extend from? It extends from the Z disc to the next Z disc. So the myofilaments within the myofibril allow for the muscles to contract. The myofilaments within the myofibrils allow for the muscles to contract. And this is the functional unit of the contraction. There are dimensional arrangements of the sarcomere. So now let's look at the dimensions. The I band contains only thin filament. This is our I band. You see, it only has actin. You see, that's why it's only one line. So they took a cross section. This cross section represents for me the I band. You see, the I band only has thin. That's why they call it the thin filament, or the thin filament is the I band. Then now let's look at the H zone. The H zone contains only thick filament. You see, these are your myosins. But the outer edge of the A band and the zone of the overlap contains a mixture. You see, it has thin filaments in blue, like here, thin filaments, and it has thick filaments as in the H zone. So now this is the sliding filament mechanism. This is very, very important. We have been talking about it, but now we're gonna put it in perspective. The thin filament slide past the thick filament to make the tension. You already know how that happens. So who slides by who? Does the thick slide by the thin or the thin slides by the thick? The thin filament slide past the thick filament. So how does that happen? There is an interaction. We know the hole and the golf head is going to go in there. The interaction of thin and thick filament. You can demonstrate the sliding filament mechanism using your hands. So your fingers are the zone of the overlap, and the inner edges of your palms are the Z disc. So you see, that's how they slide. Now, let's look here. When the contraction happens, you see, that is a relaxed sarcomere. We know that our sarcomere is always from a Z line to a Z line. Do you guys see the Z line and the next Z line? So the sarcomere is made up of two Z lines. The Z lines contract. So the relaxed sarcomere is made up of two Z lines, and the Z lines are going to contract. And now you see what happens? Now the myosin is going to make the actin slide by. That's how you see when they are relaxed, they are not touching. But when they contract, the myosin head is going to bind to the actin active site and pull it closer, shortening it in this direction. And here also, the myosin heads on this region are going to bind to the actin and pull it to the middle. So we are pulling everything medially to the axis in the middle. And that what causes the sarcomere to get shorter. Why does the sarcomere get shorter and smaller? Because the Z bands are getting, because the Z discs are getting smaller. So the I band and the H zone narrow during a muscle contraction. There's a sliding filament mechanism of contraction. So a band remains unchanged. So myosin heads grab the thin filament and pull toward the M line. So the Z disc move closer because they move closer and the sarcomere shortened. Where is your M line? That's the M line. So everybody, the thin filament on this side is pulled to the middle in the M line. And the actin filaments that are on the other side are pulled to the middle M line. That M line is what we are trying to get to in order to shorten and make the sarcomere smaller. The sarcomeres are arranged end to end within each myofibril. As individual sarcomeres simultaneously contract, the whole muscle fiber contracts. Now, the skeletal muscle fibers are electrically excitable cells. They have electrical properties of the sarcolemma. So now in the plasma membrane, remember how we drew the phospholipid heads and the tails? When we draw the phospholipid heads and the tails, so I need to have on my plasma membranes, I need to have on my plasma membrane there is an influx of, there's an influx of, yeah, sodium, Na, excellent. Influx. So when I look at my diagram, so I'm always repeating. So the more we repeat, the more we're going to get the concept. On the y-axis, I have millivolts. On the x-axis, I have time, milliseconds. So when there is an influx of sodium, is it coming from where there is a lot to where there's a little bit, or where there's a little bit to where there's a lot? Yeah, so that's the influx. So the influx is going to cause, what do we call this point? The resting membrane potential. That is minus 65. What is that? Minus 65 millivolt. What is that? That is the negative and resting membrane potential. NRMP, negative resting membrane potential. The cell is chilling, no action potential. Now, when you open, it is no more resting because there's going to be an influx. It's like little kids playing in a room. You open the door. What are they going to do? They're going to efflux. But here, the sodium, they want to come in, but they can't because it's closed. But in the action potential, they're going to open. When they open, there's an influx of sodium. And then there is an influx that causes depolarization. And many students have a problem. They think that depolarization is when we go down. They say, oh, D, going down, D. No, depolarization is when we become more positive because it's minus 65, now you go up to minus 45. That is depolarization, increasing membrane potential. Then what is the other phase that is known as repolarization? Repolarization. This repolarization is efflux of calcium, efflux of potassium. Excellent. So now we're going to have efflux of potassium. So there are electrical these are the electrical properties of the muscle. That is this kind of thing here. We talked about influx, efflux, action potential. Do we see that in the connective tissue? Do we see this action potential in the connective tissue? Do we see the action potential in the epithelial tissue? No, because they are not excitable. We only see this issue here, this action potential phenomenon, only in who? In the muscle tissue, and I know that's coming up next, but you prepare for it from now. Both of them. And both of them are excitable. They will conduct and mediate, facilitate action potential. But because action potential happens, are they going to do the same thing? If I give you $100 and I give somebody else $100, are you going to spend it the same way? No. So because nervous tissue and muscle tissue both are excitable, it does not mean that they will respond in the same way. When excitable action potentials pass by a muscle cell, they will contract. And when excitable action potentials pass by the nervous cells, they will mediate a response, integration, like a computer. We will see more about that. So the membrane potential in the cell is due to unequal distribution of ions. That's why the dead person, if you take a sample from outside and inside, do you think there's going to be more sodium on the outside than inside, like with everybody else? For a dead person, in our body, we are alive. Sodium is a lot outside, a little bit inside. And potassium is a lot inside, a little bit outside. Why is that? Why is not equilibrium? Why are they not isotonic? Why are they not the same why are they not equal outside and inside? Why? Why is the extracellular environment rich in sodium and the intracellular is not? Why is it the case? Let's say this is a cell membrane. This table is the cell. I have a lot of sodium outside. I have a little bit of sodium inside. I have a lot of potassium inside, a little bit of sodium outside. If a dead person, when I take a sample from here, they are equal. The sodium outside and inside equal. The potassium outside, inside equal. But for us who are alive is not equal. Why? Because the sodium, potassium, ATPase. If throw out 3, again, is the concentration gradient, bring in 2 potassium. Remember we said that. So the sodium, potassium, ATPase always make sure that the potassium does not equilibrate. Why? So when the action potential comes, you can have influx. You can have efflux. But for the dead person, can the dead person have action potential? No. Because when the action potential come in, tell sodium, please, influx. The sodium will say, influx, I only influx from where there's a lot to where there's a little bit. It's already equal. Because the potassium ATP is not working. Because he's dead. Because we can't make ATP. Sodium, potassium, ATPase. It's called sodium, potassium, ATPase. It needs ATP. Dead body cannot make ATP. Cannot power sodium, potassium, ATPase. Cannot power sodium, potassium, ATPase. Cannot throw back sodium against concentration gradient. Cannot throw potassium against concentration gradient. They're going to equal. Action potential come. Sodium say, I can't. We already equal. But for you, there will be action potential because your sodium, potassium, ATP is working. Except the snake bites you, the cobra. When the snake bites you, the poison is gonna damage the sodium, potassium, ATPase, and that's why you will lose. You're paralyzed. You're like you cannot have action potential. So you see. It's due to the unequal contribution of the ions near the sarcolemma. And the negative ions in the cytosol, and there are positive ions in the extracellular fluid. So the negative ions in the cytosol and the positive ions in the extracellular fluid. So the electrical potential in muscle fibers at rest is called resting membrane potential. You see what I was talking about? Resting membrane potential. How much is it? Minus 85. It is about minus 85 millivolt because the cytosol is 85 millivolt more negative than the extracellular fluid. The membrane is polarized. What does it mean that the membrane is polarized? It means that it is more negative. It is more negative on the inside than the outside. Action potentials. So action potentials, there we go. These are a property of excitable cells, resulting in quick temporary the property of excitable cells resulting in quick temporary change in membrane potential. So the membrane potential goes from negative to more positive and then back to negative resting potential again. It occurs in milliseconds. They are produced by the opening and closing of the ion channels that moves the sodium and potassium across the membrane. They're produced by the opening and closing of the ion channels that you see across the, so there are two types of gated. There are ligand gated channels. These are the ion channels used in the action potential. Does that mean we don't have thermal gated channels? We don't have mechanical gated channel or pain gated channels? We have that. But do they participate in the action potential? No. Only the ligand gated channels and the voltage gated channels. So what does a ligand gated channel mean? The ligand gated channel, what does it mean? So this channel right here, a ligand gated channel means it has a site to bind the ligand. So when the ligand bind it, that's the ligand gated channel. That's a ligand gated channel. This ligand gated channel, when it's bind the ligand, what happens? Excellent. But there is another type of channel known as voltage gated. The voltage gated, does it have does the voltage gated channel have a ligand site? No. So how does it open? When the action potential come by and hit it, like like a possessed, like the magic, when they do the magic, black magic, and the person changes. So that's the same thing. When a membrane voltage change and it strike, like lightning. Yeah, that's a good example. Lightning, the lightning bolt. When it's raining and the lightning bolt. Yeah, so that's sometimes the lightning bolt can open your door. You are inside the house and then the door opens. That's exactly what happens in the membrane voltage gated channels. In the normally is the key. The ligand is the key. So the key open the door and you go in. Like acetylcholine. What's a big example of a ligand that binds the channel and open the channel? Nope. Glucose doesn't bind the channel. That is facilitated diffusion. We talk about facility, but I'm not talking. We talk about ion channels. Yes, glucose binds its channel that mediate its passage from outside to inside because it's too big. I'm not talking about that. We're talking about ion channels. How do they open? You need a ligand like acetylcholine. When acetylcholine binds, what happens to the channel? It opens, and then the ions can go through it. But in voltage gated, like a lightning bolt, electricity, Strike the door opens. Now no key is the electric voltage. The ligand-gated channels. Ligand-gated channels open in response to chemicals called ligands. The ligand-gated channels open in response to chemicals called ligands. And voltage-gated channels open and close in response to changes in the membrane voltage, like electrical, like a thunderbolt, okay, that's gonna hit the membrane. Steps in action potential. There's resting, depolarization, repolarization. Resting, like here. Resting, depolarization, repolarization. 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 These are the three cycles. There we go. The membrane is at rest, is at resting membrane potential, and the voltage-gated sodium and potassium channels are closed. You see, both are closed. You see, that's the RMP. How much is it? Minus, what is the NRMP, negative resting membrane potential? Minus 85. And both are closed, right? Sodium is closed, potassium is closed. The voltage-gated sodium and potassium channels are closed. Then a stimulus would come and you see will open the sodium. So now a lot of sodium would influx, would get in. When a lot of sodium influx, what happens to the depolarization? It goes up to how much? You got to know these numbers. The resting membrane potential is minus 85. When you go up here, positive 30, that is the positive 30, that is the peak, of the action potential. So the membrane becomes less negative and peak at plus 30 millivolt. Then what happens then is that you see this little cap is called inactivation. You know when you put, if you're cooking a chicken on the pot and then you put a cap on top of it. So now you put the cap underneath sodium because sodium channel allowed so much sodium to come in, and then the depolarization went up to hit the max plus 30, and then you close. You see this cap where my mouse is? You see, pop, you put the cap. So now no more. When you hit the peak here, you close the cap. So no more sodium flowing in. Now potassium efflux. And as potassium get out, while the cap of the sodium closed, then you have a repolarization. We go down until we hit our minus 85 and we remain there. And this capping from underneath is known as inactivation. So when we say inactivation of sodium channel, that's known as from up, or you have to be very specific. When we say inactivation of sodium channel, what does that mean? So do we close it from the top? Do we close the sodium channel from the top? From the bottom. So you have to be very specific. Inactivation of sodium means the capping and closure of sodium ion channel from the bottom, not from the top. Action potentials move along the sarcolemma. We talked about this, and the T transverse tubules. The action potential at transverse tubule will initiate the muscle contraction. So the muscles would contract by electrical signals from the nervous system, converted into a chemical signal and back to an electrical signal. So you have electrical, obviously, that is the action potential. Then a chemical signal, you have calcium being secreted, and then the contraction, and then we relax again to go back into a electrical signal. So the neuromuscular junction, this is so important. In the neuromuscular junction, well, from the name, you have neuromuscular. So how many different types of tissues do you have? When you say neuromuscular, neuromuscular. In neuromuscular, you have in neuromuscular you have muscular and neuro. So you have neuron and you have muscle. The neuron will have swollen ends. In the neurons, you have swollen ends. And in the muscle, you have your T-tubules. In the muscle, you have your T-tubules. And the muscle is actually having evaginations, fold. There are muscle fold. Why are they folded? Why is there a folding here? Remember in the intestine, we said the intestine has a lot of fold for what? The intestine has a lot of fold for absorption, to increase surface area. We always do folding in order to increase surface area. So what is there a lot what is a lot that is found here? That is your acetylcholine receptors. These are your acetylcholine receptors. You see, these are the receptors. These are the receptors. These are the receptors. So what is here? Acetylcholine. 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 What did we say acetylcholine does? What? Acetylcholine. What does acetylcholine do? We just mentioned that, guys. No, that's the fold, increased surface area. Acetylcholine would bind the receptor outside the channel that's going to cause the channel to open. So now you have acetylcholine here and receptor here. And between them, this gap known as the synapse, the synapse or the synapse. So now when action potential comes in the neuron, you see why they call it neuromuscular junction? NMJ, NMJ. Why they call it NMJ, neuromuscular junction? Because it's a junction between a nerve and a muscle. Also known as the motor end plate. The motor end plate. In the motor end plate, you have acetylcholine. Acetylcholine that's going to bind this. This is the presynapse. Remember, this is synapse, right? And the action potential is coming down this path. It's coming down from here, from the neuron to the muscle. We don't call it the muscular neural junction. We call it the neuromuscular junction. So we begin with the neuron, and the neuron will propagate. The neuron will propagate to the action potential, to the nerve terminals, and that's going to release acetylcholine, and acetylcholine is going to bind. Acetylcholine is going to bind. Acetylcholine is going to bind the post. So this is the pre-synapse. That is the pre-synapse. And that is the post-synapse. And this is the synapse. So acetylcholine is going to bind here. And when acetylcholine binds, what does it do to the channels? Open the channel, and that causes the influx of sodium, and that's going to cause the action potential to travel down the T tubules, transverse tubules, and it's going to go to the cisterns of the sarcoplasmic reticulum. It's going to cause calcium to be secreted. Then after calcium is secreted, it's going to bind troponin, and troponin is going to pull away tropomyosin, exposing the whole, the active site, in order for the contraction to happen. You see, there we are. This is you see that the synaptic cleft, that is the presynaptic terminal, that is the postsynaptic terminal. And we see that action potential coming down the neuron is going to cause these vesicles that are full of what? What are these vesicles full of? Acetylcholine. And then acetylcholine is going to be released, and it's going to bind to the synaptic, the postsynaptic is going to bind the postsynaptic cleft. And that's going to cause an opening. That's going to cause an opening of the ACH receptor. It transmits a nerve impulse and an action potential from the neuron to the sarcolemma. It is composed of three parts. There's the axon terminal, the synaptic cleft, and the motor end plate. You see that's the axon terminal, that is the synaptic cleft, and that is the motor end plate. So the axon terminal is a swollen end. It is an enlargement of the end of the axon. They contain synaptic vesicles with filled inside them neurotransmitters such as acetylcholine. Neurotransmitters such as acetylcholine. So you see, this is the axon terminal. These are the synaptic vesicles, and this is acetylcholine. There's a space between the axon terminal and the muscle fiber, known as the synaptic cleft. Acetylcholine is released into the synaptic cleft, and it contains acetylcholinesterase. This is very important. This acetylcholine that is going to fuse out is gonna fuse out through the synapse and bind to the postsynaptic terminal. This is gonna need an enzyme called acetylcholinesterase. Acetylcholinesterase gonna cause acetylcholinesterase. Acetylcholinesterase breaks down ACH. There's an area of the sarcolemma that contains many acetylcholine receptors. It has folded. Why is that? Why is the end of the motor end plate has a lot of fold? Like I drew here. You see the fold? Why is there a lot of fold? I'm sorry? Say it, Noaka. What did you say? Why do we have a lot of fold? It's okay, say it.
More space. More space.
More space. More space. Why do we have a lot of fold? To increase the surface area, just like the intestine. The intestine is also folded to increase the surface area. Now, if they tell you, I'll give you a meter cloth that is this long, a meter cloth this long, and then somebody said, no, I want it to be like that. My 1 meter cloth, I want you to give it to me like this. What is longer? Which one is longer? If you have one really long bed sheet that is this long, and another one right next to it, but it's actually folded up like that, which one is longer? So you see that this increase the surface area because we need to have a lot of these receptors. Now, we need to bind acetylcholine to the receptors, but when the acetylcholine is there for too much, we don't want it to be, it's going to contract too much, contract, tremor, it's going to make like Parkinson disease. That's why we have this enzyme that degrade and break down this extra, this extra acetylcholine. Who's gonna destroy it and break it down? Acetylcholinesterase. So that's why in diseases like, for example, myasthenia gravis or myasthenia gravis, what happens in myasthenia gravis? What happens in myasthenia gravis, there is congenital, meaning you're born with it, absence of these receptors. There is acetylcholine, but there is no receptor to bind to. That's myasthenia gravis. But with the botulinum toxin, you know, sometimes they put pesticide, and the people that do the revolution and protest in the street, they throw gas tank at them. This gas tank, it inhibits acetylcholinesterase. So acetylcholinesterase is not working. So that means you will have too much what? Acetylcholine. And when you have too much acetylcholine, it's gonna bind to the receptors, causing too much contraction. And the too much contraction is happening in the gland. That's why the tears come out, the nose secretions, and the saliva, and all of this. Because acetylcholinesterase is not working. And then you have the botulinum toxin. The botulinum toxin is going to prevent the secretion of acetylcholine. So it's not even coming out. No exocytosis. So acetylcholine comes out with exocytosis. So you see, that's that's the motor end plate, and you see the excitation phase is the transmission of the signal. It is the transmission of the signal from motor neuron to the sarcolemma, and the action potential arriving at the axon terminal causes the opening of voltage-gated calcium channels. Causes the opening of voltage-gated calcium channels. Calcium entering the synaptic terminal causes excitosis of synaptic vesicles. You see? The action potential arriving at the axon terminal causes the opening of the voltage-gated calcium channels. And calcium entering the synaptic terminal causes exocytosis of synaptic vesicles. There you go. You see calcium entering here. You see this? This is very important. Calcium entering into the action potential. So you have a first phase known as the excitation phase, where the transmission of a signal from the motor neuron to the sarcolemma. So the action potential arriving at axon terminal causes the opening of the voltage-gated calcium channels. The calcium entering the synaptic vesicle causes excitation of the acetylcholine. Here, so acetylcholine, you see exocytosis. Acetylcholine is exiting the postsynaptic, the presynaptic cleft due to calcium entering. And the synaptic vesicles release acetylcholine into the synaptic cleft. You see, an action potential arrives at the nerve terminal. The synaptic vesicle release acetylcholine into the synaptic cleft. Then acetylcholine binds to ligand-gated ion channels in the motor end plate. The ion channels open and the sodium enters the muscle fibers. And then the entry of the sodium depolarizes the sarcolemma locally, producing an end plate potential. You see, acetylcholine is bound to binding site on the receptor, and there's a ligand-gated ion channel. You see? The ion channel open and the sodium enter. Guys, this is really important. The neuromuscular junction, you have the neuron, and that's the muscle. What happens? Number one, action potential would come. Then this is all on the quiz next week. The action potential would come, and then the action potential would cause the vesicles to open. And when the vesicles open, that would cause the release of so much acetylcholine. And then the acetylcholine is going to bind the ligand-gated channels. And when ligand-gated channels are open, sodium is going to enter, and the entry of sodium depolarized the sarcolemma locally, producing end plate potential. Then acetylcholine diffuse across the synaptic cleft and bind to the acetylcholine receptor, and acetylcholine receptors are ligand-gated channels. You see, ACH bind to ligand-gated ion channels in the motor end plate, and the ion channels would open, and the sodium would enter the muscle fiber. And then the acetylcholine ligand gated channels opening the ion channels, and the sodium would enter the muscle fiber. You see? The entry of the sodium depolarizes the sarcolemma, producing an end plate potential. And an end plate potential is a local depolarization at the motor end plate. So acetylcholine is bound to the binding site on the receptor, and there's a ligand-gated ion channel. You see, that's excitation contraction coupling. The excitation contraction coupling links the excitation and the contraction phases, where the end plate potential may lead to an action potential. So if the end plate potential open enough voltage-gated sodium channels, it will stimulate an action potential. You see, that's the end plate potential stimulating an action potential. Is the end plate potential, that's ligand-gated ion channel. And the action potential propagates along the sarcolemma and down the transverse T-tubules. The action potential would propagate along the sarcolemma and down the transverse T-tubules. It propagates as depolarization of one area membrane triggers the next sodium channels to open, triggers the next sodium channels to open. So you see, this is now the action potential is going down. You see what I was asking earlier? What does the action potential propagate down? T-tubule stands for what? I'm sorry? Transverse tubules. Then the action potential is going to pass through. Then the depolarization will stimulate a voltage sensor in the T-tubule, and the voltage sensor would physically open the calcium channels in the terminal cisternae of the sarcoplasmic reticulum, and the calcium would flood the cytosol. The T-tubule depolarization leads to the opening of the calcium channels in the sarcoplasmic reticulum, and the calcium would enter the cytosol. So now do you see? You see this is the action. What is the green tube in the middle? What does that represent? That's the T-tubule. Excellent. And who's passing by the T-tubule? See, plus, minus, that's action potential. When the action potential come close, who's blue? What is this blue pocket? Full of what? These are the cisterns full of calcium. When the action potential come next to the cisterns full of calcium, what happens? What happens now? Calcium flux out. Then when the calcium go out, there's going to be voltage-gated protein channels, and then the calcium is going to, the new action potentials are generated over and over to increase the calcium in the cytosol. So as the calcium floods the cytosol, the sarcolemma repolarizes. So you see, when the calcium floods, you see that's the calcium going in the cytosol. Then your sarcolemma would repolarize. That the sarcolemma would repolarize. In the contraction, you have the cross bridge cycle. So here, there you go. Now, when we let a lot of calcium out, you see the myofilaments slide past one another during the contraction phase. And the regulatory proteins allow myosin heads of the thick filaments to attach to the active sites on the actin of the thin filaments. And at the rest, tropomyosin would block the active sites. You see, that's the myosin, that's the thin filament, and that's the active site. The calcium would bind to troponin, and troponin has three subunits: troponin C, troponin T, and troponin I. Troponin C binds to calcium, and troponin T binds to tropomyosin, and troponin I binds to actin. So after the stimulation, the calcium release causes the active site of actin to be exposed. Okay. The binding of calcium to the troponin C releases the inhibition of troponin on the troponin T. So it can shift the position to allow tropomyosin to expose the active sites of actin. After stimulation, the calcium release causes the active site of the actin to be exposed. So the myosin heads bind the active sites when they become exposed. And the myosin head bound to the actin is known as the cross bridge, and that's what causes the sliding. We talked about that. The muscle contraction is a succession of cross bridge cycles resulting in the production of force. And myosin heads would grab a series of actin molecules in the thin filament, and it pulls the thin filament closer to the M line or the sarcomere. That's what we get to. You see your troponin? When troponin binds calcium, now it moves it away. So number one, ATP hydrolysis cocks the myosin head. So the myosin head is in the cocked position. And the myosin head would bind the actin. And then the power stroke occurs when ADP and phosphate detach from the myosin head. So myosin head would bind in the ADP form, and then the power stroke occurs when the ADP and phosphate detach from the myosin head, and the myosin pulls actin toward the center of the sarcomere. Then the ATP breaks the attachment of the myosin to actin, and that causes the hydrolysis movement. So ATP hydrolysis cogs the myosin head. When you go from ATP to ADP, myosin heads binds ATP, and ATPase hydrolyzes it. Energy cocks myosin head into the high energy position. ADP and PI remain bound. So the ATP hydrolysis cocks the myosin heads. So this is a thin filament known as actin, and that is a portion of the thick filament. That is a portion of the thick filament. The myosin head would bind to the active site on the actin at about 90 degrees to the thick filaments. So you see the myosin head would bind actin So when ADP and PI detach from the myosin head, myosin pulls actin toward the center of the sarcomere. The movement is called the power stroke and ends about 45 degrees relative to the thick filament. The power stroke occurs when ADP and phosphate detach from the myosin head and the myosin pulls the actin towards the center of the sarcomere. And another ATP binds the myosin head to break myosin and the actin cross bridges. You see, that is the ATP that breaks the attachment of myosin to actin. Contraction cycle is repeated. That is the contraction cycle. The contraction cycle is repeat. The ATP is hydrolyzed and myosin head recox and binds to the next actin molecule and the power stroke repeats. The average contraction requires the process to repeat 20 to 40 times. You see, that is the relaxed sarcomere. That is the relaxed sarcomere. We're moving closer to the M line. The cross-bridge cycle repeats, pulling the actin toward the center of the sarcomere. You see, we're moving toward the center. Now, I mentioned that botulism and Botox. These are toxins from the bacterium Clostridium botulinum that binds neuromuscular junction and blocks ACH release from the synaptic vesicle. So the toxin from the contaminated food, what does it do? Binds the neuromuscular junction and blocks ACH release. Remember, we mentioned that. Botulinum toxin prevents release from the synaptic vesicles. The toxin from the contaminated food, specially canned without proper sterilization, and causes botulinum. So the muscle is unable to contract and respiratory failure would follow. And clinically used Botox and to remove the wrinkles because it does not allow the exit of acetylcholine. It involves the stopping acetylcholine release and the breakdown of acetylcholine in the synaptic cleft and the removing of calcium from the cytosol. Acetylcholinesterase degrades the remaining ACH and the final repolarization would occur. That is the synaptic cleft, and you have acetylcholinesterase degrading the remaining acetylcholine, and the final repolarization process would happen. Muscle relaxation. In muscle relaxation, the sarcolemma would return to the resting membrane potential, and the sodium channels in the sarcoplasmic reticulum would close. The voltage-gated potassium channels would open to drive the action potential toward the resting membrane. See? The active transport pumps would move the calcium back into the sarcoplasmic reticulum, returning calcium in the cytosol to resting levels. So now we would take calcium back into the normal levels. Tropomyosin blocks the active sites, and calcium would dissociate from troponin C, causing troponin to return to the original state. You see, troponin is dragging tropomyosin back into the original state. Rigor mortis is the muscle spasm 3 to 4 hours after death, and that is because there is no ATP for the relaxation. Now, that shows you the excitation with acetylcholine trigger and end plate potential in the motor end plate. And that is the motor end plate. Then the excitation contraction coupling would happen where down the T tubules you have the action potential flowing, and then the resulting action potential in the sarcolemma would travel down the T tubules and triggers calcium release from the sarcoplasmic reticulum into the cytosol. Then you have the preparation for the contraction. calcium bind to troponin, which moves tropomyosin away from the active site of actin, and then the contraction, you would have actin and myosin would bind, and myosin undergoes a powerful stroke, and ATP detaches actin and myosin, and the cycle repeats, leading to contraction of the muscle fiber. For relaxation, the neuron stops releasing ACH, and the ACH, acetylcholinesterase, ACHE, degrades the acetylcholine in the synaptic cleft, and the cytosolic contraction of calcium returns to the resting level. And the active sites of the actin are blocked, and the muscle fibers would then relax. Any questions? Does